Welcome, Forensic Biology. This is going to be part 4 of chapter 5, Forensic Botany. Now, after this, your quiz will be on chapter 5. This is on Thursday. So you are doing this Thursday, therefore, meaning your quiz is going to be on Monday. Okay, so your quiz is Monday. There's a goof by me as far as the dates. I had switched the wrong Monday. I thought you guys had class this Monday. But anyways, you'll get the notes this week. Do the quiz on chapter 5, and then we will start our new chapter 11, entomology. So that will be our quiz in which quiz Monday, finish chapter 5, start chapter 11 next week. So this is part 4. It'll be going over pollen producers, spore producers, how pollen and spores get dispersed, and all of that fun stuff, and then also collection, finding it, collecting it, and analyzing it. So that's pretty much going to cover it, and really what does it all mean. So first, we need to talk a little bit about plants. We need to understand that plants are going to require reproduction, and there's two ways about typically how they're doing it. Asexual or sexual reproduction. Pollen is going to be considered a sexual reproduction. Spores are going to be considered an asexual reproduction. So first, let's get into pollen. Pollen producers, plant reproduction is either from non-seed plants or seed plants. Seed plants are going to include gymnosperms. That's here your cone plants, your evergreens, your firs, your plants of that type. Okay, usually they'll have either male or female cones. Female cones here look like this, kind of these wooden flowers, I guess. They're going to open up, and inside are going to contain the ovaries. And male cones being these yellowish, kind of dust-looking structures. And what's going to happen is that's going to contain the pollen. The wind is going to blow that, and that pollen then will be deposited inside the cone. Cone will then start forming a new plant for the most part. Okay, so what you're doing here as far as creating is with pollen is you're creating a pollen profile, and it's important. Pretty much what it is you're looking at the number of pollen grains found in a geographic area at a particular time of the year. Now that's going to change. As in the winter, you're going to have less because most plants are going to go dormant. So therefore, you're going to have less pollen for the pollen profile or pollen fingerprint, as it is also called. So during their life cycles, seed plants are going to produce cones or flowers. Okay, so they're going to produce cones or flowers. Gymnosperms are going to create cones. Angiosperms are going to create flowers. All right, and then the pollen is going to be uh, dispersed, and you're going to form another plant and be reproduced. Okay. Seed plants are the predominant plants in the assemblage or the collection of plants in any environment. So therefore, they're going to have the most likely plants to leave evidence at the crime scene. So these plants are going to be most likely to leave the evidence. Why? There's just more of them, simply put. So let's take a look at the structures here. Here you have what is an angiosperm. An angiosperm is a flowering plant. So these flowers here are going to be the reproductive organs of the flower. Here you have the male structure. This is the anther. This is the filament. Okay. And then here you have the female structure. You have the stigma, the style, and then the ovary here. Those combined are called a pistil. So you have female and male. You have the ovule. You have the petal here. So these structures here are going to determine or be responsible for reproduction. Okay, in angiosperms, they are the most common plants, 300,000 different species of plants in water. land, air, pretty much everywhere. So these are going to produce quite a bit of information that you're going to need to know. Gymnosperms are going to be the cones, not as much, as they don't have that much. They're going to include cycads, ginkgos, and conifers. Conifers being like your Christmas trees, your evergreens, like what you would see in like the mountains, the forest, things like that. So going on to pollination. Okay. First, we want to talk about pollination. So, how do these plants pollinate? Okay. One is self-pollination, is they're going to pollinate themselves. So if you look back at this structure here, you see female structure, you have male structure. Okay, so why can't they? Well, some can, and that's called self-pollination. Now, these plants here are not going to be as useful in forensic botany because they don't release a lot of pollen. Okay, so they don't have to. It's self-pollen. It doesn't travel as far, so therefore they don't need to release a lot of pollen, and it's not as useful. Okay, now cross-pollination is different plants. Okay, where you're going to, one plant is going to pollinate another plant. Now they're the same species, but not necessarily itself. Okay, so cross-pollination is going to create a larger pollen production. Therefore, these are going to be more useful. Some plants do both. Okay, some plants are able to do both. So therefore, once again, self-pollen plants are of little useful in forensic studies because it exists in a relatively small volume and they do not travel far. Okay, so these are the types of pollination. Now, method is pretty much how it happens. So gymnosperms are going to use air. Okay, the wind. They're going to carry the pollen from one plant to the next. So that's a wind. You also have animals and water, where you're going to deposit the pollen into water, let the water carry it. You'll have animals that are going to deposit the pollen in other plants. You see these common in hummingbirds, bees, insects, things like that that are attracted to these bright, colorful flowers. They then uh, take the nectar, or sorry, take the pollen, go to the next flower, and then next thing you know, you have reproduction, fertilization, and that flower then becomes a fruit. So wind pollinated plants are often dominant, mainly because they produce a lot. Okay, animals can pollinate flowering plants; they don't have to produce as much pollen, so therefore the wind pollinating plants are going to be more dominant, simply because they're just going to have a higher pollen content. Um, so therefore, they're going to have, because there's so much, they're going to be hard to determine a direct link. Okay, where animals here and here is going to be very specific to areas, so therefore it's going to create a link, a more specific link. So when pollinated, going to be overrepresented in collecting samples, therefore creating a less effective link to the individual, or from the individual to the place. And animals that pollinate flowers going to be more effective because there's not as much, so it's a little bit more rare, I would say. So here's an example of a bee pollinating flower. So as it goes to the next flower, it's picking up the pollen here. As it goes to the next flower, it will deposit that pollen into the plant, into the flower, and create fertilization there. Okay. Seed dispersal. I want to talk about that. That doesn't have a section in here. Seeds are going to be dispersed through after pollination happens. You're going to have then seeds going to be used. Once again, it can be the air, the land, uh, water. The most common, that being in the angiosperms, that's going to be fruit. So those flowers that you see, those flowers are going to, once they're pollinated, once they are fertilized, they are going to form a fruit of some sort, and then that fruit will fall off and or get eaten and then be deposited. Mostly, it's going to be deposited through some sort of feces. Okay, so pollen, you're going to find those in gymnosperms and angiosperms. Simply put, pollen is going to create a seed. So any plant with a seed is going to have pollen. They are the most, okay? The more you have, the harder it is to create a link to the suspect in the crime scene. The less you have, the easier it is because that pollen profile is not going to match the other areas. So spores, what are spores? Spores are asexual reproductive structures produced by a variety of organisms, that being non-seed plants, fungi, bacteria, things like that. Now, I want to understand that it's asexual, meaning it does not need a partner, but spores here are going to have all of the DNA in which it needs in order to survive. So you can actually understand what structure this is based on just the spore. Okay, with the seed and the pollen and things like that, it's hard because it's a sexual reproduction. You only have half the DNA. So therefore, spore producers are going to be able to create the actual organism a little bit better than that of the pollen. Okay, you're going to create same thing, just as you would a pollen profile, you're going to create a spore profile. Now, you're going to have a lot more spores that are going to be released simply because they're much smaller and they're easily transferred and they are overlooked by predators, making them useful. And that's pretty much going to be the same thing. The pollen or the spore profile is going to link it to a environment, and then you're going to be able to, or you should be able to use that environment to link it to a suspect and state that that suspect was there because based what was found on them, whether it be their dirt on their shoes or whatever it is, you can create and link the spore profile to them. So they had to have been there. Dispersal, for the most part, they're going to use the environment. They don't need another organism. They're just going to release it into the environment. It's a couple different ways. Wind, water, those are going to be the most typical ways. Here in mushroom, you have these slits here. Those are gills. That's what's going to release it onto the ground and things like that. And then you're going to get more mushrooms that are going to grow there. Okay. I do want to talk about one thing. I don't know if they have it in here. No, they don't. So spores, okay. Um, there are a couple things that I want to talk about as far as uh, endospores. Endospores are important to know because pretty much what happens is endospores are going to be pretty uh, gnarly structures. They're a little bit smaller, but what they do is they develop this impenetrable wall compared to other spores, and a lot of these are very caused with very bad diseases and things like that, such as anthrax and botulism, two of the most severe types of disease and um, chemicals on Earth. And simply put, it's because they have endospores, which you can't necessarily treat all that much. So that's why they're so dangerous for the most part, is because you can't typically treat them because of the endospores, which is a thick wall, and they are resistant. So that's causing that problems to treat them, is because they're resistant. They have thicker walls. They are smaller, and there's a lot more usually that are made. Okay, so here you can see on the ferns here, You can see the spores, sporangia, and the frond. That's just a simple leaf, but you have these spores that are going to produce on the underside of the frond. So this is the underside of it, and you get these orangish seed-looking things. Those are the spores. You also have a puffball fungus, which is going to eject the spores into the air. It's kind of like if you just poked it with a needle or something and it shot everything out. That's what that's going to look like. So pollen and spore identification and solving crimes. One is, like I said before, spores have both sets of DNA, so therefore you can create the actual organism. You can go all the way to the plant. You have pollen, which is usually a little bit bigger than spores, so it's a little bit harder, but for the most part, it's going to give you a radius as far as how far away that person was. So here you have a lily pollen. It talks about in your book a corn pollen as far as only traveling a half a mile. So if you have some sort of corn pollen on you, then you have been within a half mile of a corn field or a corn flower. So that limits your environment as far as where you have been. And that's pretty much what they're doing is they're going to match the corn or sorry the pollen or the spore to the plant in the profile, and that's going to determine where you've been. So going into this, we're going to move on to the next one. I want to talk about this. It's a little bit more important. So pretty much I want to understand, or I want you to understand that forensic paleontologists are going to be the ones, if possible, forensic paleontologists are the ones that are going to find pollen spores, collect pollen spores, and analyze pollen spores. Okay, they understand where to look as far as to find the pollens and spores. All right, a couple areas it's important. Soil, dirt, dust, those become usually important areas to look. They are definitely abundant at crime scenes, so collect those. Also, you can find it in other areas such as any type of paint, really any type of like lubricant, anything like that where that pollen can stick to. Artwork, paint, what else? Like grease, anything with grease. Stuffed animals, foot impressions, things like that. Anything where that pollen can pretty much settle. So those are the kind of the idea as far as where to look. Now collecting it, yeah, because you're using such small evidence, it's microscopic, it's really small. You want to make sure that you do things right. So you are, putting it and bagging it in a paper container so the moisture doesn't cause it to decompose. And then also you are pretty much cleaning your tools after every collection and using it as well. So the most common ways in which you're going to collect is via paintbrush or some sort of brush, bristled brush, where you can brush it and collect it into some sort of container, or cellophane tape, okay, where you pretty much are going to pick it up with some cellophane tape. And also what you want to do is take that collection, you're going to collect the crime scene, but also the area around it to match that. You're creating a control. Okay, so you want to control, you want to show the control, the neutral area around it, the crime scene, and then when you get a suspect, you're going to show the pollen and the spore profile of those. And if they're all three a match, it's the same area. So that's pretty much what they're going to be doing. As far as analyzing them, pretty much looking at it under a microscope, shine light on it, look at it, compare it. Now, if you want to look at the external structures, you're going to use what is a scanning electron microscope. and that's because it's going to get into a little bit more fine detail, whether it has spines or hair or whatever it may be on it. So that is going to be how you would analyze the surface or look at it. After scientists narrow their search, okay, so they're looking at it, they understand, they find it, they record their evidence, they're going to narrow their search with illustrations. What's going to happen is they're going to go to a herbarium, pretty much a place that holds a lot of documentations, records, and data of the different spores or pollens, and they're going to match them. And then once they have matched them, they're going to get an actual plant. Then they're going to take that plant and get the spores or the pollen from that, and then match them side by side. So it's the process of first of identifying what, and 100% being certain that that is that plant, finding the record, finding the database, then collecting spores and pollen from the actual known plant. That's going to be your kind of control, and then comparing them. So it's kind of a longer process than you probably would imagine, but the reason for this is, once again, we're putting this into a court case, right? So you don't want to leave any reason of doubt that that person or that pollen profile is specific to that area. So you want to make sure you hit all the right stops and do all the right things. Now, what does that evidence say? Well, this evidence says that the pollen in the spore evidence is unlikely to be the primary or only evidence in a crime, simply because it's circumstantial. This is circumstantial evidence. It does not prove that that suspect created that crime, right? What it only proves is that suspect was at that location. Now, we don't necessarily know a time. We can do a season, but we don't necessarily know an exact time. Now, you can go based off the seasons and things like that, but what you're trying to do with forensic botany is you're trying to link the suspect to the crime scene. So that's pretty much why it's not going to be the only, or it shouldn't be the only type of evidence that you're using, because it's not going to hold up. It's purely circumstantial, but is a solid circumstantial evidence when used with other types of evidence. So you would need to know more about it as well. Well, thanks. That's part 4. That's our last part of chapter 5. We will continue our studies in chapter 11 the following week. So remember, I want to tell you, your quiz is on Monday, chapter 5 quiz. Monday, you'll have a quiz, and then you'll start into chapter 11, entomology. Have a good weekend.